Hi y'all, welcome back. This is lecture 19. We did skip 18, that was intentional. And then we're just moving on to just one lecture this week because y'all are working on your paper and we have the final wrap up stuff. So I think this week one lecture, next week one lecture, and then the last week I had set aside for review and other things for presentations, also to give you some time to work on your presentation. So this is it. I have also on the Canvas site put up sort of all of the final modules so that you can see them. You can't necessarily watch the videos of everything yet because I haven't filmed the final videos, but otherwise you have access to just about everything at this point. So I want to do a little bit about epigenetics. I think this will be just one video, but I'm going to watch the time and see how it goes. And then I will see you in lab on Tuesday. Let's talk about all the weird things that control your genetics. So before you start this, go back and review what is a Barr body and what makes a Barr body barred. Like, why does it get, well, Barr was the guy's name who discovered it, but why does it get all wound up and become chromatin that can't actually be accessed? Why does it become heterochromatin? I want you to just quick review chapter 17 for regulation of transcription for eukaryotes, because a lot of that material we're going to continue on in this lecture. And then what is the structure of a chromosome? We talked about the P arm and the Q arm at one point a long time ago. I want to be able to point those things out as well as the centromere. And then let's talk about all the exceptions to the rules. So big ideas here is we're going to talk about epigenetics and the epigenome and what that has to do with turning genes on and off, silencing them or enhancing them. And then what that actually means that there's an outside control to using or not using your actual genes. I want to talk about how this can be heritable. I want to give you a few syndromes that this is tied to, with the caveat that some of you are doing presentations on diseases or syndromes that are epigenetically controlled. So I'm going to skip over that at this point and let you take that information from your peers or your own presentations. And then we'll talk a little bit about epigenetics and cancer. And I want to point out some of the databases that we use, because at this point we're developing information on epigenetics. It's not like Mendelian genetics that we've understood for 150 years. This is really new stuff that we don't know that much about. So we're developing databases. I want to show you two of them. We as in the human species. So epigenetics, and epi means above, these are controls that are above the actual genome. So this is not, in this whole lecture, we are not altering the DNA itself. We're not altering the code. What we are altering is access to the code. This can happen in tissue-specific ways. So in one type of tissue or one type of cell, I have a gene turned on that I have turned off everywhere else. It can also happen in ways like during development, as you age or go through everything from your initial cleavage and mitotic divisions, as you go from being a fertilized egg to a fetus, there's a lot of switches that get turned on and off, and roles that those cells have to go through. They control those by accessing or not accessing genes, not by having different code in different cells. The epigenome is the state of a cell and its outside controls that are allowing things to be transcribed or translated and not allowing things to be transcribed and translated. And then when I talk about an epigenetic trait, I'm saying this is a stable, meaning it's not going to change in the next couple minutes, but it's also inheritable by the daughter cells either through mitosis or meiosis. So just because you went through mitosis does not mean that you didn't pass on your genes for sure, but also the epigenetic controls that are allowing access to some genes and not to other genes in the chromosomes. And where it gets really interesting is as we get to the end of this, I want to show you how we can pass on epigenetic traits and pass them on to children and grandchildren, not just from one cell in your body, but actually to your descendants without altering the genome. Just passing on alterations of access to the genome. So the big questions really are, how do we do that? Like, how do we turn on things and off things as you go through your normal developmental process? And then how do you actually pass it on? I don't have answers to you in all of these cases. We can observe these things happening. we know that they get passed on. I don't have a great series of explanation of how the cells, I do in some cases, but not a full explanation of how the daughter cells actually inherit the epigenetic tags themselves. In some cases, we see that during the S phase, all of those tags are removed and then they're retagged. In other cases, they seem to stay with their tags, but we need to open up the chromosomes to replicate them. But those chromosomes never become accessible to RNA polymerase. So I think all these words make sense to you now. I don't know if they would have when we started. I guess the advantage of the videos is you can stop me and go back. But also, if you're taking notes and you want to email me at the time you're asking questions, feel free to do that. I am around pretty much all the time. I go to bed at 10 and I get up pretty early, but I can answer you if you have questions while you're watching this. Just shoot me an email and I can answer it. Then we have more of a dynamic conversation because this stuff gets weird. I guess it doesn't get weird. It gets unexpected. I think your great-great-great-great-great-grandparents would have kind of intuitively understood that the environment itself can cause changes to the body and that can get passed down from generation to generation. But as we've understood that we have particulate inheritance and that our genes are these little particles of DNA, it becomes harder to understand how the environment can affect those particles on a case-by-case basis. And one of the ways we know a little bit about this is by looking at diseases and syndromes that come from when this epigenetic controls or the epigenome is not acting in the way we expect. So I will add a caveat here. One of the students is doing one of these syndromes. So I'm going to skip Engelmann syndrome because that's part of her presentation. But I'm going to talk about Prader-Willi, and then later I'm going to talk about Beckwith-Weidman syndrome. And then at the very end, I'll talk about cancer development and how they're controlled by epigenetic regulation. Prader-Willi is sort of the classic go-to in epigenetic controls that are acting in an unexpected way. Prader-Willi results from either the deletion of the paternal chromosome 15, so the child doesn't get the chromosome 15 at all from its father, or the mother has two chromosomes that are 15. You inherit two of your mom's 15s and don't express your dad's. This problem is mainly caused by the fact that when you inherit this region right here of your chromosome 15, so on the Q arm, region 1, 11.2 and 12 and 13.1, this one right here, when you inherit that part, it is differentially imprinted, or what we call genomically imprinted, and I'll talk about this later in the lecture. When you inherit it from your father, you use it. You use dad's chromosome 15, but you imprint mom's. So whatever you inherit in that region from your mother, it doesn't matter because you methylate it and turn it off. You can only use your information from your father, and so if you don't have any information from your father, either your father didn't give it to you or your mom gave you 2 copies and your dad didn't give it to you, what it means is you don't have access to those genes because you only access them if they came from an egg. When you have Prader-Willi syndrome, we have a characteristic, it comes with this characteristic unbelievable hunger. Small hands, small feet, some physical characteristics. There are some cognitive issues that go with it, but the big thing is it makes you really, really, really hungry all the time. And part of the challenge that people who grow up with this is that they're constantly hungry. They gain a lot of weight. Honestly, the newer drugs that are like Zemprec have had some success in treating people with Prader-Willi because it suppresses those hunger urges. So there's a kind of aside to nutrition. And then this disorder is a syndrome caused by imprinting. It is not something we can cure. We don't have a drug for it because you're missing access to part of your chromosome 15. You have it, you just can't use it. Epigenetic changes are outside above the actual genome, and they actually occur through 3 major mechanisms. And we've talked about all of these already. I just want to summarize them as part of this class or this lecture. They can occur through methylation. Methylation is when you add methyl groups, usually to a cytosine, but we've been adding them to histones and some other parts. I want to talk about all of that. If you add or remove methyl groups, you can, usually with methyl, if you add them, it decreases the amount of transcription, or you can remove them and increase the amount of transcription. You can modify histones, you can methylate them, acetylate them, phosphorylate them. I have a little bit more detail here about how we regulate the use of the DNA that is wrapped around that area of a histone by modifying the histone proteins itself. And also we've talked briefly about there's large regions of RNA that your body is producing that are not coding. They're not controlling for protein production, but they are acting as switches and suppressors and enhancers and silencers that actually give you more or less or none access to your genes and make those proteins. So I want to go back to methylation, which I think we have covered now 3 or 4 times. This idea that you can add a methyl group, and it's here, but basically a CH3 group to functional group to a molecule, you methylate it. And when you do that, that changes the access of that area of the DNA code to things like RNA polymerase to make a transcript with, or it causes it to bind up so tight that it's actually never accessible to RNA polymerase. This is reversible. In some cases, those methyls can be removed, and that causes the molecule to become more accessible to the RNA polymerase. Methylation takes place after DNA replication. So if you go all the way back in your head to mitosis, meiosis, when it was January, and we talked about the fact that during the S phase of interphase in DNA, and again later when we talked about DNA replication, the methyl groups can be removed and then reattached during cell differentiation or reattached during other parts of the life. We also see removal of methyl tags as cells differentiate. So as it goes through the process of becoming a new type of cell, maybe you start out with a stem cell and that cell gives rise to many different kinds of cells. Those descendant cells that come from the progenitor, the parent cell, might have different methyl tags or have had their methyl tags removed compared to the parent. The enzyme here that does this methylation is called DNA. It's working on the DNA. Methyl, it adds a methyl group. Transferase. And I know we mentioned in class the other day, and I feel like in our class and in a couple classes, it's all kind of coming around for me, where this becomes important over and over again in biology. Ase means enzyme. And then this is a Latin sentence, so you read it kind of backwards. This is an enzyme that transfers methyls on DNA. DNA methyltransferase. DNA methyltransferases is a group of enzymes that basically add methyls to cytosines, and they act on, and I'll zoom in on this in a bit, areas where there are CG dimers. So that means, do it this way, areas of the genome where there's C's and G's together. They're all often in repetitive sequences. Let's see if I can do this without losing track here. So I have, this is a dimer, this is a dimer. Dimer means two mers, two monomers. Dimer. They'll methylate the cytosines in these groups. And then when those cytosines are methylated, we add a methyl group or a methyl functional group. It becomes 5-methylcytosine. This acts differently. It has a slightly different polarity. It wraps up the DNA in a way where these methyl cytosines tend to bunch together because they are less polar than the original cytosines and the original guanines. They snuggle together to avoid the polarity of the other molecules, and it makes the DNA there less accessible. There are 3 types of DNA methyltransferases that I want you to think about, and only because I want you to think about why we need more than one type. There is DNA methyltransferase 1, or 1. I think it's probably dynamite 1. Maintains the established patterns of methylation. So what this does is if we are going through from one cell to the next cell, it's making daughter cells. We maintain the same methylation as the parent. We use DNA methyltransferase 1. There is DNA methyltransferase 3A and B. They actually are used when we are instituting a new methylation pattern. So something is becoming methylated for the first time in a cell, and then its descendant cells will inherit that. When the descendant cells inherit a methyl that their parent cell has, they'll use the DNA methyltransferase 1 to maintain that. Methylation is reversible. We can add or remove methyl groups. I want to summarize here. This takes place on cytosines that are next to guanines. These are called CpG dinucleotides, or dimers. These dinucleotides are clustered in little islands near promoters. So what that means is I have little clumps of sequences of DNA that are lots of CG, 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 CG. There's a C and a G together. This is a great place for methylation. They're frequently found in long pieces of C's and G's near the promoter. Now I want you to think about, what does a promoter do? And I definitely want to ask you on the final exam as well. Promoters are on the DNA. And they're where RNA polymerase attaches in order to transcribe or make an mRNA transcript. If the promoter is unmethylated, and basically it's not all twisted up because it's bound together with all of these things that are slightly less polar than the world around it, so it doesn't twist up and lock up. If the promoter and the island in front of it is unmethylated, we can transcribe. If the promoter is heavily methylated, this area becomes sort of inaccessible. That means that the promoter region is not accessible to RNA polymerase, and we cannot transcribe the genes. If they are methylated, they are silenced. If we have, and it's important, and we kind of talked about this in bacteria, but not in eukaryotes, if you have an island of C's and G's, these methylated areas, next to essential housekeeping genes, they are always unmethylated. Housekeeping genes are genes that you need to make all the time. If they are genes that we need to turn on and off during the lifetime of the organism, we can methylate them and demethylate them, possibly. What that looks like on a molecule itself is, here is the DNA. This is the phosphates and the rails of the DNA. Here is the rungs of the DNA. So here's a cytosine and a guanine. Here's a guanine and a cytosine. And when we methylate this, it causes it to twist together. This is the major twist. It causes a groove in the DNA. This is now inaccessible to enzymes that want to copy the DNA and turn it into RNA. We also have methylation where we methylate areas that are going to be heterochromatin or permanently inaccessible in this chromosome. This is something like what happens to the centromere. The area around the centromere is generally completely methylated. It is an area where we don't have active genes. What the methylating does at that point in the centromere is make sure that it's very difficult so that you can't translocate easily, so it protects that area of the centromere. We also see this in playing a role in X chromosome deactivation. This is making the bar body in mammals. And mammals that have two X's, one of those X's is bar-bodied. It's bound up and made inaccessible to anything like RNA polymerase, so that it's never used. Some of the way that it is bound up is by having a series of methylations, and then it's completely turned into heterochromatin. Heterochromatin is where it's not usable. If you remember, euchromatin is usable or accessible. When I cause a region that was once euchromatin to be methylated on its cytosines or its CP dimers, I can actually cause a specific region to have a specific pattern of expression. I want to talk about this in a minute. I can methylate on different chromosomes, different areas, depending on who is the parent that gave you that chromosome. So we have parent-specific regions of methylation, and we also have regions that are randomly methylated. For example, the areas on the chosen X chromosome, one chromosome is kept in each cell, one X chromosome is randomly methylated, and then we make some of this long non-coding RNA is expressed by that bar-bodied chromosome, and it converts it, it keeps it as a bar body all the time. It's this schist long non-coding RNA. And this is something we talked about, it's been about a month since we talked about bar bodies, but X and Y regulation of different gene expression. We can modify the histones. I feel like we've done this a couple times in different lectures. You know that the DNA is wound around the histones. And as the histones, they have a set of 8 histones inside a single nucleosome. Of those 8 histones, their little N-terminus, so the N side of their amino acid or their amine side of the end of their amino acid chain is sticking out. And so we can acetylate it, we can methylate it, and we can phosphorylate it and change how we actually are accessing and expressing those genes. So I want to add one thing here. I want to talk about how we recognize where and identify the location of histone modification. When I am going to modify a histone tail that's hanging out, I modify histone 2, either at part A or B. I modified histone 3 and histone 4. And you can see that's what's happening here. I'm looking at those different modifications. I also want to point out that we can have a kind of a shortcut for what kind of functional group is attached to the N-terminus. A P basically means, and I have it down here too, is a phosphate group is added, it's phosphorylated. An Me, is a methyl group is added, or it's methylated. And an AC is an acetyl group, acetyl. I'm adding an acetyl group. So when I modify those histones, I can go back here. That was really, really fast. I'm going to leave this at the bottom. There it is, the functional group abbreviations. I am going to, when I modify my histones, I'm going to modify it at 4 possible locations. Histone 2 at either A or B, histone 3 or histone 4 of my 8 histone nucleosome shape where the DNA is wrapped around it. So when we name where are we modifying something, we have to talk about what histone is involved. I need to talk about what amino acid is actually being modified, and then the position number of that, and finally a little code for what functional group did I really ask. So I'm going to shortcut this, and I'm going to say that my methylation, oops, I spoiled it already. My modification occurred at H4A7Me. I'll give you a second to figure out that the blanky blank, the amino, so this is going to be an amino acid, at position of histone number has a functional group attached. And just so you know, over here, this is a chart of the different amino acids. I tend to use the 3-letter code, but there is also a 1-letter code. So if you're trying to figure out what this A means right here, A stands for alanine. You ready? So H4A7Me means that the alanine, I don't need to write it, the alanine at position 7 of histone number 4 has a methyl group. I'm not trying to make you crazy. It actually makes this easier to talk about, but it's kind of like learning a new language at the end of the semester. I just want to introduce it to you because I know that this is going to be something that you are going to talk about again. I think so. I think this is something you're going to talk about again. I also would point out that this is something, and I know I've said it in the last couple lectures, that we are learning more and more about. This is super relevant to our treatments of cancers in all mammals and possibly other species, but also it's treatment to a lot of the aging diseases that we see. This is something we're learning to understand and going to need to be able to, in 10 years, to really talk about, because it's going to, I think, probably play a major role in how we understand medicine in the future. Histone modification, we're winding our DNA around the histones, and we're adding functional groups to those histones to change the rate or whether we can express or not. We talked about a couple weeks ago, maybe 2 weeks ago, that HAT, histone acetyltransferase, opens up the structure. We add an acetyl group to my histones, and that causes it to relax. When that happens, my DNA in between and around my histones become more accessible to transcription. If I remove those acetyl groups, the enzyme that does that is HDAC, histone deacetylase, or an enzyme, let's do it this way, an enzyme that takes my acetyls and dees them, that means removes them, deacetylases, on my histones. It actually causes the genes to be silenced. Adding the histones makes them more accessible. Removing the acetyls from the histone makes them less accessible. This code is something we're trying to understand. When I look at my entire genome, I am trying to understand how the histones are regulated to add accessibility. As in the slides, we talked about how they slide apart and slide back together. How do we regulate during our lifetime and during development all of the accessibility to all of these genes? It's so much more than something like a bacteria that just has to have something blocking the operator. We've got to be able to modify this over the course of an entire lifetime, and also cell by cell basis, so different tissues need to act in different ways. A couple more pieces, and I want to describe this but leave the words pretty simple, because I'm really not going to ask you too much, but I think you should have in the back of your head how these little guys are working. These are miRNA. This MI stands for micro, so they're really tiny pieces of RNA. They're not coding. That means that they don't actually make proteins, but they do get involved in regulation. When your body makes miRNA, you make a sequence of RNA that has coding that kind of, it binds to itself. So it makes these little hairpins where it forms, this is hydrogen bonds between matching nucleotides all along the edges of it. I don't know how well you can see this. These things, when they travel through the body, they bind to mRNA. And as they bind to mRNA, this little piece on the end cracks off. And because these guys are now not held as well together, they unravel. And one side binds to a matching side of the mRNA. In plants, it's usually a nice match. In animals, it's a not as nice match. But what they do is then start to block translation. So they make those mRNAs less usable by the ribosome and may actually help those mRNAs degrade. We think they have a role in breaking down that polyacetyl tail at the end of the RNA. And when the RNA is missing that polyadenated tail, so I've been saying acetyl all day, that polyadenated tail, the enzymes in the the cell itself start to break it down because they no longer recognize it as a usable piece of mRNA. If you want, this is a really good article in the Wikipedia, actually, that is appropriate level for us explaining how micro mRNA works. Long non-coding, we always write it with a lowercase l, but I made it uppercase here so you could remember that it was an L. Long non-coding RNA regulates gene expression similarly to how other smaller pieces of RNA regulate gene expression. We've already seen this in the regulation of the Bar body. That's part of what the XIST, the gene, regulates. It makes its own long non-coding RNA that convinces the Bar body to stay heterochromatin and not be accessible. Long non-coding RNA looks like mRNA. It gets a cap and a tail. So it has that 5' cap and it has a polyadenated on the 3' side. It also gets its introns spliced together, or introns spliced out, its exons spliced together, but they don't code for anything. What they do is they help regulate codes in other places. We can bind together with other pieces of coding mRNA or DNA and regulate the accessibility of those usable pieces of code. Changing topics, not that much, but kind of changing from RNA. I want to talk about a little bit what happens when you only need one allele. In X chromosome deactivation, when we make a Bar body, one of the reasons we make that Bar body is because every person only needs one X. But that's a very different process than the fact that on your chromosome 15, some of the regions you only need one allele. 2 alleles is too much information. So in places where you only need one allele's worth of information, we have areas where we have monoallelic, one allele gene expression. This is shortcutted as May. I have trouble recognizing this as a word. I always use monoallelic and then gene expression here, but monoallelic expression. And like conceptually, this is kind of an easier topic, I think, than the last one. You don't need 2 copies of every gene in order to be a normal functional human. For several of your genes, particularly things that regulate growth and development, you only need 1 copy. 2 copies is too much. There are 3 ways that this is regulated. Either depending on which parent gave you the gene, you use one parent and not the others. So I'm going to show you some examples of imprinting or genomic imprinting, where you only use one parent and you silence the other parent's information for your whole life. You can, however, pass it on, and depending on whether you're mom or you are dad, your children may inherit the gene that you had silenced. So kind of very similar to how we talked about when we talked about expression in chloroplasts. It can be one or maternal inherited genes. It can be one generation removed that you see expression. It can be random. This is something like the X chromosome. It just randomly, it doesn't care which parent it came from. It randomly just deactivates one of the genes, either on the X chromosome and sex chromosomes or on autosomal genes. I love the story of genomic imprinting. I love how we understand it. I think it will take you a little bit of thinking. When we imprint, and I'm talking about genomic imprinting, because imprinting can mean a lot of different things. Genomic imprinting means you only express one gene, and it depends on which parent gave you the gene. We see this really commonly in mammals and in growth factors. In mice, and this is the classic study, and there is more here. If you click on this link down here for epigenetics imprinting, it takes you to this whole story on a click-through story to talk about these mice. There are 2 genes that we understand that are regulated in mice. We think this is similar in humans as well. The gene for insulin-like growth factor and the gene for insulin-like growth factor's receptor are both regulated by epigenetic monoallelic expression. They're both regulated by imprinting. The one mouse that if you develop into a normal mouse, you get both of these genes, 2 alleles for this one, for insulin-like growth factor, and you get 2 alleles for the receptor. One from mom and one from dad. But if you're going to develop into a normal mouse, you use mom's receptor gene and silence dad's, and you use dad's hormone gene, insulin-like growth factor, and silence mom's. If you get a gene and you inherit a gene from mom that is good for the insulin-like growth factor and dad that is broken, that means that you cannot make it. If you inherit a gene from mom that is broken and dad that is good, you're fine. But you are carrying one broken and one good. If you, in this case, are dad, you have a real possibility of passing on bad alleles to your offspring that actually get expressed, because they're going to express your dad. So whatever you pass on to your offspring, they're going to express. And you have a 50% chance of passing on the wrong, broken one. We also see this in ligers. So if you've ever seen a liger, it's a lion-tiger crossbreed. It's a hybrid. Ligers get really, really big. That's because in lions, the growth factors are, I might be backwards on this, but I think that in lions, the growth factors from the father are silenced and you express the mother's. If you are a tiger, it's the other way around. When a liger is born, it's the result of a lion dad and a tiger mom. And that means that you get 2 alleles, both which are usable about growth factor. They both get activated. So a baby liger actually gets twice as much growth factor as it's supposed to get, and they get really, really huge because they're not imprinting either gene because the instructions are not clear from their parents. This is also a really, really big problem when we talk about doing things like IVF and trying to understand how to do artificial reproduction, because we don't necessarily pass on all of those genetic imprinting codes in the way we think. And we sometimes have problems with children developing during IVF because they didn't get the right imprinting instructions, because this is reprogrammed every single generation. During gamete formation, both chromosomes have their imprints erased from their original, whatever you pass on. So you pass on something that is usable, whether you could have used it or not. And then the pattern of methylation is determined by whether it came from a sperm or an egg. That means it matters who your parents are, and then you decide whether you use those genes. In humans, Beckwith-Wiedemann syndrome, which is one we talked about at the very beginning, is a result of abnormal imprinting. This is not the only cause for this. So if you look this up, there are multiple reasons why you can develop this syndrome. But one of the causes is an unexpected methylation of chromosome 11 on the P arm at location 15.5, where the maternal copy of the gene is either not accessible, it's been methylated, or the baby has 2 copies of the paternal gene. You need mom's gene at that location in order to actually function. You methylate dad's. So it doesn't matter if you have 2 good ones from dad's. If you don't have a good one from mom, you're going to develop BWS. If you have a bad one from mom, even if dad gave you a good gene at that location, it doesn't matter because you methylate dad's genes there. Kind of onto bar bodies again. When we talked about bar bodies, this is a random inactivation of an entire chromosome. It doesn't matter whether you are looking at dad's or mom's X that you got. If you have 2 X's at a certain point during early development and you are a mammal, one of those X's in every cell is deactivated, and that deactivation is passed on during mitosis to every single cell that depends from that cell. So it effectively silences about 900 genes on the inactive X, but that's good because you don't need 2 copies of all of those alleles. That inactivated X stays inactivated in every cell that comes from that cell lineage for the life of the organism. The only exception is that when gametes are generated, and so you're making eggs, and you're going to pass on one X or the other, you don't pass on a methylated bar-bodied version of an X. You demethylate them, they unravel just like all the other X chromosomes in the body that are actually accessible, and you pass on one or the other, but they're both usable. Throughout your entire genome, monoallelic genes are scattered across most of your somatic code. So in your non-sex chromosomes, in your autosomes, there's a whole series of scattered genes that you only need 1 copy of. We are just starting to understand these and the control mechanism for turning them on and off. Again, this is a problem if you are making babies using IVF. We may not be getting all the code in order to turn the right genes off at the right time and methylate in the right order. We also have a problem with cloning mammals because of this as well, because often when you're cloning, you don't get a code to turn one side off because there's no dad in that case. There's only mom. And it's interfered with our understanding for a very long time of how to clone mammals, and often why it just doesn't work for us. I want to skip on to cancer. I pulled this out of this is a meta-analysis of a bunch of different types of methylation, hypo and hypermethylation that leads to cancer. Hypo is not doing it enough. When we look at cancers, we find that hypomethylation, not having enough methylation, is a hallmark of every single cancer that we've examined. Now, this is the first step, not all the steps, but there does seem to be some really strong links to environmental factors like smoking and pollution and stress that lead to low levels of methylation that can then trigger long-term changes in the body that lead to cancer. This was first noticed in the 1980s in colon cancer. And we look at cells in the colon that are actually cancerous, they don't have the expected levels of methylations of normal colon cells. So if you want to pause it here or read this, fascinating stuff. I am on the edge. This is getting to where I'm not sure I can explain it well, much beyond sort of the genetics intro idea, partially because cancer isn't my thing. If cancer is your thing, as a side note, I highly recommend The Emperor of All Maladies. It's a phenomenal history of how we understand cancer and where we're going. They start to talk a little bit about the epigenetic code as he gets to the end of the book. The first time I read it, I was kind of on board with the idea that by the time he finished the book, we were going to cure cancer. And of course, we didn't because he's telling a story that's still happening. But absolutely amazing summer read, for real, if you're interested in some of the controls of cancer here. Mutations in the epigenome seems to also be tied to cancer as well. Those mutations means that things do not or get methylated in multiple sequences in a way that we don't normally have in those cells. Often what we do is we silence one allele of a gene that is normally a tumor suppressor. So it gets too methylated. And so that gene that is normally looking out for you and making sure you don't develop tumors gets silenced. And then a second mutation activates an oncogene, or a gene that stimulates mitosis. And those two factors together lead to unregulated cell division and no tumor suppressor watching out for you. So I have a little sub table for this because I think it's worth thinking about this pattern. Is if we turn off your tumor suppressors, maybe we hypermethylate it so we have a high level of methylation so that you cannot access your tumor suppressors, and we turn on genes too much that do mitosis. Onco means cancer. These are mitosis genes, however. Cancer is just you doing mitosis in ways you're not supposed to do. I can often stimulate, this is a pathway to developing cancer. And then when I look at overall this instability and unpredictability from your body's point of view of what is methylated, I'm rearranging my chromosomes, I'm accessing some genes inappropriately or in unexpected places. That's a recipe for developing cancer. The BRCA1, this is breast cancer 1 gene, is hypermethylated. It is a gene that is supposed to be a tumor suppressor. If it's hypermethylated, that means it's inactivated, and it leads to the development of breast cancer, ovarian cancer, and other soft tissue cancers. So the combination of mutation, what it's talking about here, is that you inherit one good BRCA gene. This is a good one. And you inherit the other allele is a bad one. It's something's wrong with it and it doesn't function. You already have a really high chance that all you need to do is make one mistake. You don't get the backup that people who have two good versions of this, you don't get the backup. When this goes wrong, now you are no longer looking around those tissues for mitosis gone wrong. We also have the CDKN2A mutation in bladder cancer. I think the BRCA1 is more recognizable for you, but if you want to look at the list of things that we know are involved in hypermethylation that are supposed to be regulating normal mitosis and making sure you're doing mitosis correctly, I have a list of the genes that we know regulate that and what happens when they do not work. Broken result. We do have drugs in development for this. They are first generation drugs. We do know that epigenetic changes are often reversible, right? You can demethylate something, but we don't want to just randomly start demethylating things because sometimes you don't want to activate a gene. So our first generation, and they are still coming through development, focus on reactivation of specific regions that we know have been incorrectly methylated. Hopefully later on, we will be able to use a second or third generation drugs that will mean that I can target specific types of cancer and I can have fewer other drugs involved in my cancer treatment. Maybe ultimately not have anything like chemotherapy. I can actually target those cells and tell them to stop misbehaving. But we're not here yet, but we are here. And we do have some cancers that we are able to target experimentally by targeting the epigenome. There's a ton of different environmental causes that affect how you use your genes. We talk about stress a lot in the modern world. Stress absolutely changes the regulation of how you regulate things like your relaxation hormones. We also rate how you regulate your circulatory system, your nervous system. We know all those things are tied to stressors in your daily life. It's a really good example of how your environment can actually affect your health. Your environment can also affect whether or not you can make proteins. And one of the weirdest discoveries in the last, 30 years, I think personally, is the discovery that epigenome can be heritable. So this is no changes to your DNA, but you can pass on those methylated tags in some cases. And when you do that, that means that your children express the result of your exposure in the environment. And one of the best studies here, this is, it's in, most of it is in Dutch, but I did find part of it in English. Absolutely fascinating. The video is in Dutch, however, of what happened during 1944 to 1945. At the end of World War II in Europe, the Germans were starving out the Dutch, and there was a huge famine. People were down to eat with rations, and they were occupied by the Nazis. People were down to rations to 1,000 or less than 1,000 calories a day. So the people that were living in the Netherlands at the time were starving to death. When that happened, pregnant women, I have here, pregnant women during that famine had children who had been gestating during that famine. Those children, when they developed in a post-war boom, so by the 1960s, food abundance was way back, and so there was a delay there, but the food abundance was back across Europe. Those children who were born in the 40s had a really high incidence compared to other children their age of diabetes, coronary heart disease, and obesity. We think they experienced epigenetic changes in the uterus due to the fact that their mother was starving. And basically they're better at getting nutrients and retaining nutrients, which in an abundant world is not necessarily the greatest thing. But the even weirder thing is their children, the grandchildren of the pregnant women during the war, so people my age, also show these epigenetic traits, also show an altered metabolism, also show higher levels of diabetes. And when we look at their genome, they've inherited from their parents because of their mother's starvation during the war. They've inherited a different regulation of how they express genes due to their metabolism and food. We see this in mice. So this is heavily experimented on. If you take mice and you lower their protein diet and you give them less and less of the methylation precursors in their diet, it actually causes permanent changes in genes in their offspring that actually expresses in their coat color. So super easy to measure. Yellow is the standard color. Agouti is, I guess it kind of means mottled, or brown and gray and brown, yellow. Agouti is what happens when they don't have normal nutrition. It causes methylation that affects a specific gene that is involved in mouse coat color. My favorite of the heritable epigenetic stories, although I have a lot because the whole thing just blows my mind, is and I have you here. I want you to go here, and I've linked it in my canvas for you as well. I want you to go and read through Lick Your Rats. There used to be a simulation, but it doesn't work anymore. It used to be you could lick your rats and watch what was happening. But at least read through and watch the little videos of how we understand the heritability of stress. In rats, the style of its maternal care, so what mom does, is passed on to its offspring. If mom is happy and she feels safe, she spends a lot of time licking her pups. If mom is scared and she spends a lot of time looking out for danger, she spends less time licking her pups. As mom licks her pups, and she does a lot of licking, the more licking actually increases the serotonin in the babies. It makes them happy, and it increases the acetylation and decreases the methylation of a gene, the GR gene, that has to do with stress. This is a less stressed rat. Alternatively, this mom, because she does not have time to lick her babies because it's dangerous, these guys stay methylated. They decrease their amounts of histone acetylation, and these guys are more stressed. And so you're like, hey, this is just genetics, right? But it turns out if I take this pup here and I give it to this mom when it first is born, this mom licks more, and this pup ends up being less stressed. And if I take this pup and I give it to stress mom, this pup ends up being more stressed because he gets licked less. When they grow up, less stressed rats lick their pups more. More stressed rats lick their pups less. It's hard to say that, licks their pups less. But what happens is this is an epigenetic control based on the situation. If you're a happy rat and you don't have anything to worry about, chances are your babies don't either. Maybe you live in a pet store, you're somebody loves you, and that you're really well taken care of. These guys don't have to worry at all, and they can focus on making more rats and living happy lives. If you're a stressed out rat, maybe you live in Paris and you don't work at a restaurant, And so you have to take care of your babies all the time, but also defend against cats. It's advantageous for this guy to be a little bit stressed and hyper aware, because that means he's going to avoid danger more often and be more on the lookout for cats. These are situational changes that affect an entire lifetime of these rats, but are based on access or not access to genes that change their behavior and allow them to sort of optimize how they live without having to wait for evolution to take a role in this. We're learning about this. These are just kind of vocabby things. I just want to show you a little bit about the databases. There are two major databases where we are tracking worldwide research on epigenetics. The National Institute of Health Roadmap of Epigenomics project is basically tracking health and healthcare outcomes related to the epigenome, and trying to figure out, one, how we can monitor them, and then develop an understanding of how treatment for diseases that are caused by epigenetic changes will occur. And the Human Epigenome Project, with his governmental and privately founded, is more interested in creating an understanding of the entire human genome and epigenome to serve as a standard for what is normal or wild and what is not wild. And these are huge ideas, kind of like the Human Genome Project or the Human Cancer Project. We need an idea of what the wild type standard human looks like in the genome and the epigenome if I want to figure out whether this situation that I'm looking at right now is normal or abnormal. And then the third step after that is, how do I make everybody's lives a little bit better from that? I have a little bit of practice with me. Practice with me. I think this will help you with the homework. So which of the following statements does not accurately complete the statement? So which is wrong? An epigenetic trait. I'll give you a second. Okay, I'm going. An epigenetic trait is not a change in the DNA sequence. It has nothing to do with your genome. It's about accessibility of your genome, which is totally the opposite of what I told you at the beginning of this class when we were starting the idea that your genes made you who you were. Yeah, they do. But also how you can or cannot use them. It's kind of like if I have a whole bunch of books over here, I have a whole bunch of books over here, and they influence the way I think and understand the world. I have access to them, but if I don't actually read them or use them, they don't really do anything for me. If you have genes and you can't access them, they're methylated or heterochromatin, they don't do anything for you, but you can pass them on. Longer question. C. elegans is a nematode worm. It's a big white worm. It's really well studied. It makes an incredibly good organism and we talked about it a million years ago. It doesn't have methylcytosine, so it works a little bit differently. But maybe they have methylated adenine. We don't know if methylated adenine is the same thing as methylated cytosine. We don't know if it serves the same function, but let's assume for this question that it does. Which one makes sense, A, B, C, D, if methylated adenine acts like methylated cytosine? I'll give you a minute. I actually only had 3 good possible options here. The first one, please don't pick the first one. But you can pick the second one. Removing methyl groups increases gene expression, because in our bodies, adding methyl groups to cytosines reduces gene expression. Removing methyl groups from cytosines increases gene expression, and we're expecting those to have the same pattern. If I'm guessing that C. elegans kind of works like us, but not exactly the same. Methylation silences genes. But that's my last question for you. One more big lecture after this. I think you can probably see why I really want you to have the lecture on the epigenome. This stuff is fascinating, and it is definitely where we are going with genetics. We understand the code, but the code doesn't work the way we thought we did because we can't always access parts of it, and maybe accessing just part of it works better than accessing two pieces of it. One more layer of complexity makes genetics, I don't know, more fun? I don't know, although you have told me I don't understand the word fun. More fun for me. I will see you in class. The lab is, you've probably already seen me in lab, but the lab booklet is also in Canvas there, and there's a couple videos to watch, a little bit about amoeba sisters and a little bit about licking your rats. And I'll see you in class. All right, bye-bye.